evening all. We'll just wait for a few more minutes to let others join and then we can start today's session. Hello everyone, welcome back to week 5 for the PMRF sessions of NPTEL online course Drug Delivery Principles and Engineering. So we'll start with some of the key points that have been discussed in week 5 lectures. Feel free to stop me at any point if you like. and we can discuss your doubts further. So, we have been talking a lot about various drug delivery systems for the past few weeks. We have looked at, first of all, what is a drug delivery system, why do we need drug delivery, why do we need to study drug delivery in the first place, and so on. And then we started looking at actual drug delivery systems, wherein we learned about what is a prodrug, why do we need prodrugs. Then we talked about polymer drug conjugates and what advantages does this system have. Then we started talking about reservoir systems, wherein we learned that if you have your drug at a very high concentration at the core of this drug delivery system, and if it's surrounded by a non-degradable membrane, then the drug will, through diffusion process, will slowly be released in its surroundings. So that was your reservoir drug system. Then we learned about metric systems, wherein instead of just having the drug concentrated at the core, it is dispersed throughout the metric system, and that was another drug delivery system we saw. We also learned about what are liposomes, what are their advantages, what are their disadvantages, and we also talked about hydrogels. So until now we have seen various different drug delivery systems. So this session is a bridge between drug delivery and tissue engineering, which we will be discussing in further detail in the upcoming weeks. So in this week's lectures, we have learned about what is protein adsorption, and first of all, why protein adsorption, why do we need to study about this phenomenon. So what exactly is protein adsorption? So this is wherein, so we all know what proteins are, right? These are polymers made out of amino acids. Our body has a lot of proteins. So these proteins act as weak surfactants. So what does this statement mean? So proteins are they have both hydrophobic as well as hydrophilic parts, right? So what are hydrophobic parts? They are nothing but the part of the protein which hates water. So it will try having hydrophobic interactions. It will try staying away from water. On the other hand, the protein also has parts which are hydrophilic, that is water loving. Let me just so because of this tendency, because of having both hydrophobic and hydrophilic parts, these are what do proteins do? Whenever they encounter any surface, they will try adsorbing on the surface. Adsorbing meaning they'll just attach themselves on the surface. So why this happens is because, as I said, proteins will have hydrophobic parts, right? So first of all, why does this become important? Why are we learning about protein adsorption? Is let's say you have an implant. You want to put this implant into the patient's body, and once you do that, what happens is, since the implant will have a surface, most of the proteins in the patient's body will try adhering to this surface, because they want to, as most of the surrounding of this implant is going to be a lot of water. So the proteins will then, what would the proteins do? Since some part of the protein is hydrophobic, that hydrophobic part will try excluding itself from the water, and they will try attaching on the surface. So that's why this particular thing becomes very important in drug delivery. So what happens if you see this particular point? Whenever a material is placed in a patient's body, first of all, it's going to interact with water, since there is abundance of water surrounding that implant, and then the proteins are going to start attaching to the surface because of their property. And then what happens is, so you have water, then you have proteins adhered on the surface of the implant, which is then then the cells in the body will try attaching themselves to these proteins, because a lot of cells have surface receptors for these proteins, and ultimately what is going to happen is your implant is going to be covered with cells, And then, so why protein adsorption becomes important? Because what kind of cells are going to attach to your surface is governed by what kind of proteins are first of all adsorbed on this surface. So this becomes very important because then this could lead to immune reaction, this could lead to rejection of the implant and so forth. So as I was saying, cells have proteins known as integrins. So what are these integrins? These are transmembrane proteins. So they have some part in the membrane, they have some part outside this, they have some part outside inside the cell as well. So whatever receptor or surface protein the cells have, they bind to this, they get absorbed on this biomaterial because of their interaction with the proteins. So this can be harmful in some cases, but this phenomenon can also be useful for us. For example, we want a lot of cells to adhere to our material. For example, let's say you're trying to put an implant in a patient's body, and this is a bone implant, and you want a lot of cells from the patient's body to adhere to this implant. So in that case, what you can do, there are some proteins which allow cell attachment. So what you can do, you can pre-adsorb this surface, pre-adsorb the implant with these proteins, and then you put it in the patient's body. So what will happen, because of the protein layer that is already present on this implant, a lot of cells will start attaching themselves to this implant. So this depends on the application. In some cases, you don't want these cells to attach to your implant. So in that case, you can put certain, you can modify the surface in such a way that most of the proteins will not be able to adsorb onto the surface. So this was something that was discussed in week 5 lectures. Then the lectures start going towards actual tissue engineering. So what does tissue engineering mean? It is nothing but using cells, materials and engineering principles to improve or replace biological tissue. So you must have heard a lot about this, right? Making artificial heart, making an artificial kidney. So first of all, why do we need tissue engineering? Is because there's a lot of anomaly in between demand and supply. So a lot of people need organs because they might have been gotten into an accident or their organs are failing. So there are a lot of patients who are always in demand of healthy organs. But on the other hand, there are not many human donors available. So that is one reason why we need to learn tissue engineering, because if we are successful in artificially making organs, then it would be a great thing in medical field. We would be able to replace failed organs, or we would be able to easily make these organs available to a number of patients rather than just harvesting them from human donors. So that is one thing. And also, even if we right now see that there are few donors available, there is always a chance of disease spreading, because maybe the donor is already having some disease which was not diagnosed before, and now that you've taken, let's say, kidney from such a particular donor and you have implanted it into a patient's body. Now we don't know what diseases the kidney might have already been harboring, so there is a chance of disease spread. And also the immune reaction part, right? You must have heard about this a lot of times. So we need to thoroughly check whether, since, for example, let's say I am looking for an organ. Let's say I am looking for a kidney transplant. So now, let's say my very good friend is trying to donate her kidney to me, but in that case, we both are two different humans, right? So my body is not going to be very well adapted to somebody else's kidney. So there are always chances of immune rejection, and this is one of the reasons why organ donation or organ replacement fails. So all of these reasons then make it important that we need to know how tissue engineering is done, what are the principles of tissue engineering, and how very well we can make artificial organs. So all of these points were already discussed in this week's lectures. And then there are various ways of doing tissue engineering. We can either generate the whole tissue in vitro meaning outside the body in the lab. So using this tissue engineering principles, we can generate this whole tissue and replace the damaged tissue, or we can generate part of the tissue in vitro and then replace only the damaged portion. So this is sometimes done for liver transplants, right? Not so liver has a lot of regeneration capacity. So instead of replacing the entire liver, what people will do is they'll just generate this part of the liver in vitro and then replace only the damaged portion. Or what else can we do? We can put scaffolds to regenerate part of the tissue in vivo, and then or we could add supporting material to support the damaged tissue. This is very well done in case of bone implants, or you must have heard about hip implants. So these are the points that were discussed in this week's lecture. So with this knowledge, then we can move ahead with discussing some of the assignment questions. Is it okay with everyone? Am I audible to all of you? I am assuming that you people can hear me. You can hear me, right? And also the screen is visible to all of you. Can somebody just confirm this? Yeah, thank you. Okay, so we can move on towards some of the assignment questions then. So the first question is, which of the following cells do you think should be commonly used in a recellularized scaffold for skin tissue regeneration? And we have to choose two correct options. So if you people know the answer, please let me know. You can unmute yourself and let me know the answer, or if you want, you can also write in the chat box. Does anybody know the answer? I'll wait for a few minutes and see if anybody has a response. If no, then we can discuss the answer. Okay, somebody is answering as option 1, T cells. Okay, you have to choose two options. So if you have chosen one, if you want, you can choose another option as well. Thank you for answering. Anybody else? let's look at the answers then. Is there any other response? Okay, somebody is saying 4 and 6. That is fibroblasts and 1, 2, 3, 4, 5 neutrophils. Okay, thank you for answering. Anyone else wants to give it a try? Let's look at the answers. So the correct answers are fibroblasts and keratinocytes. So what is the question saying? So we have to choose which would be the best possible cells to be used in a recellularized scaffold for skin tissue regeneration, right? So all of the other cells, if you see here, you have T cells, B cells, macrophages, and neutrophils. All of these are immune cells, right? So somebody is trying to do skin tissue regeneration. We don't want immune cells in this. We want cells which are already present in the skin tissue. These are nothing but your fibroblasts and keratinocytes. Hepatocytes are nothing but liver cells. We don't want them in the skin tissue regeneration, right? So the correct answer is fibroblasts and keratinocytes, because these are the cells that make up the skin tissue. So what is recellularized scaffold? So as I was saying, right, let's say you have some you have a donor, so you want to put this implant into the skin tissue into some other patient. So in that case, what you have to do is you first have to remove the cells from the donor person, because those cells would be viewed as a foreign material by your patient's body. So you don't want any immune reaction to happen there. So you first have to decellularize the implant and then add cells from the patient's body. So this is known as recellularizing the scaffold. So this recellularization for skin tissue should be done with cells which are already present in the skin tissue from the patient's body. So these would be nothing but your fibroblasts and keratinocytes. So if you start putting T cells, B cells, they are already immunogenic in nature. They would start then attacking. They would start having reactions against the patient's body. Is that clear? Do let me know if you have any doubts with the explanation or with the question itself. If yes, then we can discuss the question all over again. If not, then I'll move on towards the next question. Is that okay with you people? Okay, thank you for responding. I'll move on towards the next question then. Next is proteins denature on biomaterial surfaces. What factors play an important role in this process? So we were just talking about how proteins absorb onto the biomaterial surfaces, right? And then they denature. So if you people know the answer to this, please let me know. Again, we have to choose two correct options. Yeah, so the question is what factors play an important role in deciding how much denaturation will happen to a protein once it absorbs to biomaterial surface. If you know the answer, please let me know. I'll wait for a few minutes for you people to read through the options. Okay, someone is answering 2 and 7. 2 is biomaterial core properties and 1, 2, 3, 4, 5, 6, last, protein stability. Okay, thank you for answering. Anybody else has any other opinion? We'll discuss the answers in at the end of few minutes. Others want to give it a try? Okay, someone is answering one and two, first and last. One is biomaterial surface chemistry and mutability, biomaterial core properties. Okay, somebody else has answered first and last. Surface chemistry and mutability and protein stability. Okay, anybody else? Thank you all for responding. Anyone? Okay, I guess no more responses. Let's look at the correct answers. So the correct answer is option 1 and the last, I would say. So I think somebody answered first and last. Yeah, you are correct. So the factors that play an important role in protein denaturation on biomaterial surfaces is first of all your biomaterial surface chemistry and mutability. So you must have seen this slide in the lectures if you have followed. So proteins denature. So proteins first of all are not very highly stable. So that is your last option here. So how much denaturation will happen to a protein depends upon what is the stability of that protein. Some proteins are very highly stable, so even if they encounter a surface, very less amount of denaturation will happen to them even if they absorb on the surface, while some proteins are not very stable. So in that case, a lot of denaturation will happen to such a particular protein. So in that case, your protein stability is going to govern how much denaturation will happen. So you have your first answer there, and then if you would have followed with few more slides in the lecture, you would have come across this, wherein the denaturation will depend on biomaterial surface chemistry and water wettability. So if you have a very hydrophilic biomaterial surface, what is going to happen? So the general structure of a protein is that the hydrophobic part of the protein is buried inside in the core of the protein, while on the surface you'll have the hydrophilic groups. I'm sorry. Yeah, so this particular general structure is because the proteins present in our body, they always have aqueous environment around them. So to avoid the hydrophobic interaction with water, the hydrophobic part is buried inside, while the hydrophilic amino acids are always on the surface. Now, in case you put a hydrophilic biomaterial surface inside the body, let's say your implant has a very hydrophilic biomaterial, in that case, what is going to happen? Since proteins already have hydrophilic part outside, they're just going to absorb on that. Not much denaturation will happen. But if you have a very hydrophobic surface and you put that in the patient's body, in that case, what is going to happen? Because now your surface is very hydrophobic and the hydrophobic part of your protein is buried inside, that hydrophobic part will try interacting with the hydrophobic surface. In that case, the protein is going to completely flip out. The hydrophilic parts will go inside, hydrophobic part will come outside, and then that will get absorbed to the surface. So in that case, a lot of denaturation will happen to the protein. Its complete structure is getting distorted, right? So how much denaturation is happening will depend upon, first of all, your protein stability, how stable your protein is, and then the surface chemistry, whether your biomaterial surface is hydrophobic or hydrophilic. So the only two correct options for this particular question are biomaterial surface chemistry and metabolity and protein stability. If you look at other options here, biomaterial core properties, they have nothing to do with protein absorption. Atmospheric pressure, amount of dissolved helium in the solvent, amount of nitrogen in the atmosphere, partial pressure of oxygen in surrounding atmosphere, none of these matter when the implant, when your surface is already inside the patient's body and there are proteins all around it. So is this clear? Are there any doubts in this question? If there are doubts, then I can go over the explanation once again. If not, then we can move on towards the next question. People who are joining a little late, it might be, you might have missed the initial discussion, but we have just started with the assignment questions and we are on second question right now. And we'll move on. If anyone wants me to repeat the previous questions in case you have missed it, I can do that at the end of the assignment. But for now, we can, I guess, move on. If anybody has a doubt, please let me know. Okay, let's move on then. So the next question is, pre-adsorbing of cell adhesive proteins such as blank and blank increases attachment of cells to the biomaterial surface. So we have to choose two cell adhesive proteins from the given options here, which will help in increasing the attachment of cells to the biomaterial surface. So if anybody knows the answer, please let me know. Anyone wants to volunteer for this question? Okay, somebody is answering second and third, fibronectin and collagen. Okay, thank you for answering. Anyone else has another response apart from option 2 and 3? Okay, somebody else is also saying option 2 and 3. Thank you for answering, both of you. Anyone else would like to give it a try? Okay, I guess no more responses. Let's look at the answers. Yes, the correct answers are option 2 and 3. So both of you are correct, whoever is answering. Fibronectin and collagen. So I was talking in the introductory slide, right? That so so this phenomenon of protein absorption to the surface could be of great advantage to us if we are trying to pre-absorb, if we want a lot of cell integration to happen with our biomaterial. So in that case, to increase this particular property, what we can do is we pre-absorb cell adhesive proteins, which two examples of these are fibronectin and collagen, to the biomaterial to increase the attachment of cells to the biomaterial surface. And in case we don't want this attachment to happen, there are other examples of proteins which are non-adhesive, non-cell adhesive. They actually they don't let cells attach to the surface. They'll try decreasing this attachment of cells to the surface. So one such example is albumin. So which one to use depends upon what application you are looking for your biomaterial. So for cell attachment, we have cell adhesive proteins such as fibronectin and collagen. Those are the only two options in this particular list of options here, which are cell adhesive proteins. Is that okay? Is this clear? Nothing much to explain here. These are just two proteins which are very well known for their cell adhesive properties. Any doubts here, or shall I move on? Okay, let's look at the next question then. Next is, which of the following is an example of tissue engineering? Again, we have to choose two correct options. So I'll give you people some time to read through this list of options. If anybody knows the answer, please let me know. Okay, someone is saying option 1. So you have to choose two options. Somebody has answered as option 1. Another person is saying option 1 and 3. Okay, thank you for answering, both of you. Anybody else has any other answers? So when you will be giving your final exam for this course, please make sure you read the questions correctly, and wherever it says two correct options, just go for, you have to select two answers. Others would like to respond to this question? Okay, somebody saying 1 and 4. Okay, thank you for answering. Anybody else? Okay, let's look at the answers. So the correct answer is option 1 and 3. Whoever answered that, you're absolutely right. So tissue engineering is nothing but it could be generating whole tissue in vitro and replacing damaged tissues in patients. We just talked about it at the start, right? Or it could be generating only part of the tissue in vitro outside the body and then replacing the damaged portions of the tissue inside the patient. It could be putting in scaffolds to regenerate damaged part of the tissue in vivo. So what you do is, instead of just putting the entire tissue into the patient's body, you take the scaffold. Scaffold is nothing but the ECM matrix that would be present around the tissue. You just put that into the patient's body and let the patient's body heal itself. The cells will then start integrating into the scaffold, and then they'll start building the tissue bit by bit. Or it could be adding support of filler material to support the function of the damaged tissue. So all of this comes under tissue engineering. So if you look at the options here, first option is correct. Growing cells on a plastic surface is not tissue engineering, it is just cell culturing. Then you have adding support filler material to support the function of damaged tissue. Yes. Culturing larvae for silk, no. Tissue engineering, this is not what tissue engineering is. Collecting spider silk for webs, no. Integrating scaffold in cancer tissue so that it allows higher proliferation of cells, definitely not. Culturing primary cells ex vivo and culturing immortalized cells. So all of some part of these are nothing but techniques people use to study cells. Tissue engineering is wherein you are actually trying to rectify a damaged tissue in a patient's body. So only option 1 and 3 are correct. Everything else is not an example of tissue engineering. Is this clear? Do people have doubts here? If there are doubts, I'll wait for a few minutes. If not, then we can move on. next question then. Next is adsorption can be, again we have to choose two correct options. We just discussed this at the start, so should be pretty easy to answer. Anybody wants to give it a try? Okay, somebody is saying 2 and 3. Temporary surface phenomenon as well as permanent surface phenomenon. Okay, thank you for answering. Anyone else has a different opinion other than option 2 and 3? Thank you for people responding in the chat box. Okay, somebody else also saying 2 and 3. Thank you. Anyone else? Okay, let's look at the correct answers. Both of you are correct. I guess two people. Oh yeah, others are also answering 2 and 3. Yeah, so all of you are correct. The correct answer is temporary surface. So adsorption is nothing but, first of all, adsorption is this surface phenomenon. It is, first option is already gone. It is not a bulk phenomenon. That is known as absorption and not adsorption. It is a temporary surface phenomenon. It could be a temporary or it could be a permanent surface phenomenon depending upon how stable your protein is, and it's not driven majorly by gravitational forces. That's an incorrect option. So this is what we were talking about in the introductory slide as well. So what is adsorption? It is defined as a partitioning of a chemical species between a bulk phase and an interphase. So always remember, adsorption is nothing but it is a surface phenomenon. It could be permanent or it could be temporary depending upon what kind of species is absorbing to the surface. If your protein is very stable, it will have very less interaction with this particular surface. It could have a temporary attachment to the surface. In case your protein really likes the surface and there are no other forces which are trying to remove that protein from the surface, in that case it could be a permanent surface phenomenon as well. Is this clear? Okay, I guess there are no doubts here. We'll move on towards the next question. Next is, what is not a strategy to reduce protein adsorption on biomaterials? Again, we have to choose two correct options. So please read the question carefully. It is asking what is not a strategy to reduce protein adsorption on biomaterials. If anybody knows the answer, please let me know. Okay, people have already started answering. 4 and 5. Increased roughness impart positive surface charge. Okay. Others? Thank you for answering. Okay, let's look at the answers. All of you are correct. The correct answers are option 4 and 5. Increase roughness and impart positive surface charge. So we were just talking about this, right? So if you want to reduce protein absorption on the biomaterials, what you can do is you can use hydrophilic polymers, because if there are if your surface is hydrophilic, as I was saying, since proteins have their hydrophilic parts exposed on their surface, very less amount of interaction, the denaturation of the protein won't be much. In that case, very less amount of proteins will try attaching to your hydrophilic polymers. So if you want to reduce protein absorption, one strategy is to use hydrophilic polymers, or you can coat with PEG. PEG is nothing but polyethylene glycol, a polymer. It has this branch structure, and if you put PEG on the surface of a biomaterial, it just acts as a brush and it just repels the proteins that are trying to attach themselves on the surface. So that is another strategy to reduce protein absorption. And then you can also pre-adsorb with albumin. Not very a lot of proteins won't like to interact with albumin, so you coat your biomaterial surface with albumin. So these three are strategies to reduce protein absorption. The question is, what is not a strategy to reduce protein absorption? One would be to increase roughness. So if you have a very rough surface, it gives the proteins more surface area to attach themselves. So if you have a rough surface, you'll have more protein absorption. Similarly, if you have a charge over your surface, most of the charges on the proteins will then try interacting. If you have a negative charge, positively charged proteins will attach themselves on the surface. If you have a positive charge, negatively charged proteins will try attaching themselves on the surface. So your last two options, which are increase roughness and impart positive surface charge, they actually don't help in reducing protein absorption on biomaterials. So these are the two correct options. Is this clear? there any doubts here? If not, then I'll move on towards the next question. Next is the phenomena of displacement of kinetically favored proteins by thermodynamically favored proteins to adsorb on a surface is called what? So if you people have understood and studied the protein absorption part in these lectures, this should be a pretty straightforward answer. Please let me know if anybody knows. If not, then we'll discuss. Okay, somebody is answering. I think 2 people are answering option 4. Okay, Roman effect. Anybody else wants to give it a try? Thank you for answering. Okay, most of you are saying option 4. Alright, let's look at the answer. All of you are correct. The correct answer is option 4, Roman effect. So what is this Roman effect? So what happens is, so there's competitive absorption, right? So first of all, whenever the biomaterial surface is exposed inside the body, what will happen is, so there are various different kinds of proteins in a patient's body. So, and most of these proteins, they could be in a very high concentration, others could be in very low concentration. So what happens is, whenever a biomaterial surface is introduced in a patient's body, kinetics first of all takes the first major role. So the proteins which are present in a lot of abundance, proteins which are at a very high concentration, they are their numbers are very high. They will first of all rush to the biomaterial surface, and they will first of all get absorbed. They'll form a layer over the biomaterial surface. As time goes on, what happens is, even though these proteins are abundant in nature, their interaction is not very strong with the biomaterial surface. Later on, what happens? Other proteins which have very strong attraction to the surface, they might be very low in number. That is why they were not able to first of all initially attach, but they have very strong attraction. So what they do is, they try displacing the first layer of the proteins, and then they attach themselves to the biomaterial surface, because they have a very strong adsorption to the surface. So the first phenomenon was kinetically favored, because as it depended upon which protein had the highest concentration. So more number, they immediately took over the surface. That is the kinetically favored protein absorption. Later on, what happened? The more stable ones, the proteins which had very high adsorption to the surface, they then started adsorbing. So this was thermodynamically driven. So what happens is, whenever a protein's biometal surface gets exposed, first of all, there will be a layer of kinetically favored proteins which would then be replaced by other proteins which are thermodynamically favored. So this entire phenomenon is known as Froment effect. So this is a pretty straightforward answer to this question, but you should be knowing what is happening when a material surface is put into a patient's body. So this effect is known as Froment effect. Is this clear? You can also read about it in this slide. This is essentially what we just discussed. If there are doubts with this explanation, please let me know. If not, then I will move on. Okay, let's look at the next question then. Next is for transplantation in humans, allografting is one of the most common approach. What are the major drawbacks of this approach? Again, we have to choose two options. I'll give you people some time to go through the question as well as the options. If anybody knows the answer, please let me know. Okay, people have already started answering. Somebody is saying option 1 and 2. Lack of donors, spread of disease. Okay. Anybody else would like to give it a try? Okay. Others are also answering as 1 and 2. Anybody else has a different opinion other than option 1 and 2? Okay, let's look at the answers. Both of you are correct. Has anybody else answered? Okay. Both of you are correct. The correct answer is option 1 and 2, lack of donor and spread of disease. This is what we were discussing in the introductory slide as well. So transplanting from one human to another human is first of all known as allografting. So what are the disadvantages? So this is the only practice that we have right now, right? So to give organ transplant. So what are the drawbacks of this method? First of all, there is a very huge anomaly between supply and demand. We have very less donors, but more number of patients are always waiting to get organs. And also the problem of the risk of spread of disease, which we discussed at the start, that if the donor was carrying some disease which was not getting screened, which we couldn't identify during the screening process, and if this such an organ gets transplanted into the patient's body, there is a chance that they might get the disease might spread to them as well. So instead of solving one problem, we have generated another problem. So this is a very serious case. This shouldn't happen. Sometimes happens. That is why this is a drawback of this method. And also immune rejection. So half-life of organs is only around 10 to 15 years. After that, the body will start giving a very strong reactions against transplanted organs. So that's another problem. So allografting, the major drawbacks of allografting are lack of donors and spread of disease. Is this clear? Okay, let's look at the next question then. Next is, which surface is more favorable for protein absorption and why? So we have been talking a lot about protein absorption right from the start. So this should be pretty easy to answer. Again, you have to choose two options. So if anybody knows the answer, please let me know. Anyone would like to volunteer for this question? Okay, people have started answering. Second and fourth, hydrophobic surface and rough surface. Thank you for answering. Anybody else? No more responses? Okay, let's look at the correct answer. So yeah, whoever answered in the chat box, you're right. The correct options are option 2 and 4. So we were just talking about this, right? So absorption of proteins on the surface. So if you have a rough surface, first of all, you'll have a lot of surface area. So first of all, proteins like interacting with surfaces because they don't like being in water, since their hydrophobic parts are not compatible with the water around them, so they are always looking for surfaces. So the moment you put any biometal surface, most of the proteins would like interacting with this. Rougher the surface, more the surface area, more amount of proteins could absorb onto the surface. So the favorable surface for protein absorption would be a rough surface because of its available surface area, and also more hydrophobic the surface because it is energetically more favorable for them to displace water. They don't like interacting with water, so they will displace, so they will try attaching themselves, absorbing themselves on the surface. So more hydrophobic the surface, more would be the protein absorption. Similarly, more rougher the surface, more protein absorption. Is this clear? I think that's all the questions I had for you for this assignment. If anybody has a doubt in any of these questions, I can go over the explanation again. We can discuss it in detail. If not, that's all I have for you for this meeting. If people have missed any questions at the start, you can let me know and we can discuss the initial questions again if you want. If not, that's all I have for you today. Does anybody have any doubt? Or would like me to repeat some of the initial questions? To let me know. For this is for people who have joined a little late. I can wait for a few minutes here and take up your questions. If not, then we can meet next week, next Tuesday at 6 o'clock. Others who are staying back in the meeting, do you have doubts? If not, then I can end the meeting here. Okay, if that's all, then I will stop the meeting. Thank you all for joining and see you next Tuesday at 6 o'clock. Is it okay if I stop the meeting here? Okay, thank you for joining.